Good morning, students. Welcome back to your biology class. Today, we will have the revision on the cellular respiration or the two steps of cellular respiration. The first step is glycolysis and the second step is Krebs cycle or TCA cycle. So, let us start with the first step. Glyco comes from glucose and lysis comes from breakdown. So the word glycolysis means the breakdown of glucose. Introduction. Glycolysis is the sequence of 10 enzyme catalyzed reactions that converts glucose into pyruvate with simultaneous production of ATP. In this oxidative process, 1 mole of glucose is partially oxidized to 2 moles of pyruvate. This major pathway of glucose metabolism occurs in the cytosol of the cell. Cytosol means cytoplasm. This unique pathway occurs aerobically as well as anaerobically and does not involve molecular oxygen. Well, next, the phases of the glycolysis. Glycolysis leads to breakdown of 6-carbon glucose, that is the C6H12O6. So 6-carbon glucose into 2 molecules of 3-carbon pyruvate, with the enzyme catalyzed reactions being bifurcated or categorized into 2 phases. Phase 1, preparatory phase, where ATP are required to start the process, and phase 2 or payoff phase, where ultimately ATP is synthesized. So the first one, preparatory phase, it consists of the first 5 steps of glycolysis, in which the glucose is enzymatically phosphorylated by ATP to yield fructose 1,6-bisphosphate. And then this fructose 1,6-bisphosphate is split in half to yield 2 molecules of 3-carbon containing glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this is the preparatory phase where previously prepared ATP are required to start the process. Now moving on to the next step, that is the payoff phase. This phase constitutes the last 5 reactions of the glycolysis. This phase marks the release of ATP molecules during conversion of glyceraldehyde 3-phosphate to 2 moles of pyruvate. Here 4 moles of ADP are phosphorylated to ATP. Although 4 moles of ATP are formed, the net result is only 2 moles of ATP per mole of glucose oxidized, since 2 moles of ATP are utilized in the phase 1. So ultimate production is 4 ATP and 2 ATP are required to start the process, and 2 is the net result. Now TCA cycle, also known as Krebs cycle, coming from the scientist discovered Hans Krebs. TCA cycle essentially involves the oxidation of acetyl coenzyme A to carbon dioxide and water. And this tricarboxylic acid cycle is the central metabolic pathway. And this cycle is the final common oxidative pathway for carbohydrates, fats and amino acids inside the cell. So now what is the definition of the TCA cycle? Citric acid cycle or TCA cycle or carboxylic acid cycle or Krebs cycle essentially involves the oxidation of acetyl coenzyme A to carbon dioxide and water. And what is the location? These reactions are occurring in the mitochondrial matrix in close proximity to the ETC. ETC is the electron transport chain, which is the last step of cellular respiration but not included in your syllabus. So students, you can see over here the entire Krebs cycle, but don't worry, no need to practice this diagram for your exam. Just see, acetyl coenzyme A is now transforming into citrate, then isocitrate, then alpha ketoglutarate, then succinyl coenzyme A, then succinate, then fumarate, then malate, and the final is oxaloacetate. And this is the entire cycle of the TCA. Now, what is the significance? Complete oxidation of acetyl coenzyme A. ATP generation, of course, which is the main target. Final common oxidative pathway, because this procedure releases the ultimate product, that is the ATP. Integration of major metabolic pathways. Fat is burned on the wick of carbohydrates, and excess carbohydrates are converted as neutral fat. So this is the entire cellular respiration. Thank you.